Chapter 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then go on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population, that is, formed by the growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Next topic, cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other consequence of the cell, and eventually divides into two daughter cells is termed as cell cycle. Although cell cycle in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Topic: Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cell in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can process through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase and M phase or mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle in a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to separation of daughter chromosome, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase or gap1 phase, S phase, synthesis phase, G2 phase or gap2 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell has diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same, that is 2n. In animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in the preparation for mitosis while cell growth continues. Some cells in the adult animal do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further exit G1 phase to enter an inactive stage called quiescent stage of the cell cycle. Cell in this stage remain metabolically active but no longer perfoliate unless cell on to do so depending on requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honey bees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants, chapter 3, identify plant species and stages at which mitosis is seen in haploid cells. Topic: M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. Though, for convenience, mitosis has been divided into four stages of nuclear division. Karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following four stages: prophase, metaphase, anaphase, and telophase. Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase. In the S and G2 phases, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which has undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called esters. The two esters, together with spindle fibers, formed mitotic apparatus. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed, and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids, which are held together by the centromere. Small disc-shaped structures at the surface of the centromere are called kinetochore. These structures serve as the sites of attachment of spindle fibers formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatids connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as metaphase plate. The key features of metaphase are spindle fiber attached to kinetochore of chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase plate, those spindle fibers to both poles. Anaphase. At the onset of anaphase, each chromosome arranged at The metaphase plate is split simultaneously and the two daughter chromatids, now referred to as the outer chromosome of the future daughter nuclei, begin their migration towards the opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed toward the pole and hence at the leading edge, with the arms of the chromosomes trailing behind. Thus, anaphase stage is characterized by the following events. Centromere splits and chromatids separate. Chromatids move to opposite poles. Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows the following key events. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete element. Nuclear envelope develops around the chromosomes cluster at each pole, forming two daughter nuclei. Nucleolus, Golgi complex and ER reform. Topic: cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, karyokinesis, but the cell itself is divided into two cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by the relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral wall. The formation of the new cell wall begins with the formation of a simple precursor called cell plate that represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria, plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytism. Example: liquid endosperm in coconut. Topic: significance of mitosis. Mitosis, or the equational division, is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cell of the upper layer of the epidermis, shell of the lining of the gut, and blood cells are being constantly replaced. Mitotic division is the meristematic tissues, the apical and the lateral cambium, result in a continuous growth of plants throughout their life. Topic: Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior, that is, leptotene, zygotene, fiscitine, diplotene, and dikinesis. During leptotene stage, the chromosomes become gradually visible under the light microscope. The compaction of chromosomes continues throughout leptotin. This is followed by the second stage of prophase 1, zygote. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrograph of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptamal complex. The complex formed by a pair of synaptic homologous chromosome is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived compared to the next stage, that is phycytine. During this stage, the four chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called recombinase. Crossing over leads to recombination of genetic material of the two chromosomes. Recombination between homologous chromosome is completed by the end of phycytine, leaving the chromosomes linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptamal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the site of crossovers. The X-shaped structures are called chasmata. In oocytes of some vertebrates, diplotins can last for months or years. The final stage of meiotic prophase 1 is dikinesis. This is marked by termination of chasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Prophase 1. The bivalent chromosomes aligned on the equatorial plate, the macrotubules from the opposite poles of the spindle 5 attach to the kinetochore of homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at the centromeres. Telophase 1. The nuclear membrane and the nucleolus reappear. Cytokinesis follows and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended stage of the interphase nucleus. The stage between the two meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2, as much simpler prophase than prophase 1. Topic: Meiosis 2. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosome aligned at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochore of sister chromatids. Anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosome which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell by sorting of macrotubules attached to kinetochore. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of dyad of cell, 4 haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which conversion of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms, even though the process per as paradoxically result in reduction of chromosome number by half. It also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution. Thank you.